Drug degradation during dissolution testing. This is the topic for present video, and the topic is important because if the drug get degraded after release in the dissolution testing, then dissolution testing will not give you the accurate results. Dissolution testing is a performance testing for formulations. Dissolution testing is related to the bioavailability, and dissolution testing is a in vitro test to predict the in vivo performance of the formulations. Dissolution testing is a performance test and is a quality parameter and directly related to safety and efficacy. So, accurate dissolution test results are much important and required. If the drugs get degraded during dissolution, then the dissolution results will get hampered. If drug degradation happens, the degradation will affect the results accuracy. So, whenever the dissolution method is being developed, that time it is required to be studied that whether the drug is getting degraded or precipitated in the dissolution medium. Generally, proper selection of medium pH and buffer is a important parameter, or you can say it as a key parameter for preventing the degradation. If you know that the drug is acid labile, that time you can skip the acid pH and acidic pH and you can perform the dissolution in neutral or basic pH. Select the proper buffer or buffer composition so that the degradation can be prevented and you can rely onto the dissolution results accuracy. But if still the degradation occurs, then some composition changes are required in the dissolution medium. Generally, sometimes antioxidants are used, sometimes chelating agents are used, sometimes specific buffers are prepared, and then the dissolution is tested. So, if the drug undergoes oxidative degradation during dissolution testing, then the antioxidant like BHA, butylated hydroxyanisole, can be used. It is a synthetic antioxidant and it prevents the oxidation of the drugs. BHA is added in the dissolution medium and it prevents degradation of active pharmaceutical ingredients by inhibition of the oxidation reactions. It ensures stability and reliability of dissolution test results. BHA can prevent the oxidative degradation and that's why it maintains the drug's potency. It ensures consistent dissolution profiles, provides reliable data, and protects the API during testing. So you can rely on to the dissolution results. BHA concentration or level in the medium is required to be optimized during method development. And BHA is used for those drugs which are sensitive to oxidation, and BHA can enhance the overall stability of the dissolution process. Other antioxidants or other chelating agents which can stabilize the dissolution media or which can stabilize the drug in the dissolution media and prevent degradation are like EDTA. EDTA prevents oxidative degradation by neutralizing free radicals and metal ions. Adding the antioxidants can extend the reliability and duration of dissolution tests. Oxidative degradation of drugs. So sometimes as an example, you can consider this slide. Drug immediate release film coated tablet. During dissolution testing, oxidative degradation observed. The degradation led to reduced drug amount at later time points. And this happened because of the oxidative degradation. The root cause Fenton type reaction was there. It was caused by iron from the film coat and peroxides in the media. So you might be knowing that in the film coating of tablets and uh, in the film coating of some pellets, the colors are used. Those colors are ferric oxide colors, and those ferric oxide colors contain iron. Polysorbate 80 is also called as twin 80 as a brand name, facilitated this reaction leading to formation of harmful free radicals. These radicals contributed to oxidative degradation of the API. So sometimes these smaller things can affect the drug degradation and can have impact onto the dissolution. So mitigation strategy can be by eliminating or the by finding out the key elements contributing to degradation. Polysorbate 80 and peroxide buildup, iron catalyzing penton type reactions. Reduce the peroxide levels in polysorbate 80 by managing lot sourcing and limited uses to 30 days. Use ethylene diamine tetraacetic acid, that is EDTA, to chelate the iron ions and prevent radical formation so that the degradation can be prevented. EDTA binds to metal ions and prevent Fenton reaction. Many times the EDTA level or concentration is required to be optimized. Sometimes you can refer the literature available onto the dissolution media with EDTA for specific molecules, and you can use those concentration as a starting point. EDTA and other oxidants can improve the stability of API or drug substance in the dissolution media and can ensure the more reliable dissolution results. When you are testing for the stability, consider adding antioxidant to prevent degradation caused by the oxidative stress. And results from these enhanced testing methods will be more reflective of real-world drug performance. One point I wanted to add here, like what happens when the drug get degraded in the media. So initially you will observe the release, but on later time the release will get decreased, and this is why, because the drug is degrading in the media. For example, if you are working on IR formulation and if it is a containing highly soluble drug, you are performing in acidic media, so the drug will get released within 15 minutes. For example, 100% release in 15 minutes, but when you test the same at 30 minutes, you will not get 100%. You can get 80% or 90% based on the degradation. So that time you need to justify that the decrease in the dissolution at later time point is because of the degradation. And also in the dissolution method development, the application of the drug stability data can be given. Force degradation data can be given. Mild degradation data or degradation under mild acidic conditions can be given and supported for justification. So whenever there is a degradation in the dissolution media, in the dissolution testing, that time the media in which the degradation is happening, that should not be used, because it will not give the correct results. So drug degradation in dissolution testing is of important thing, is of much importance, and justification will be required. You can use antioxidants like BHA, BST, EDTA in the media. You can also change the dissolution pH and you can select the buffer composition, ionic composition, and based on that the dissolution testing can be performed. I hope this video will help you to understand this new information and degradation during the dissolution testing. Thank you for watching the video, and let's meet soon with such kind of informative video.